So in our last lecture, we started talking about cell theory and very basic cell structure. What I want to talk to you about today is more detail about the cell structure. The cell is a really beautiful, complex part of our bodies, and today we're going to go into more detail about those different parts. We're going to be learning about prokaryotic cells, eukaryotic cells, and then the organelles that are found inside of eukaryotic cells. In this lecture, just like in the last lecture, there will be quick questions for you to consider, something to help you think about the information that we'll be talking about. So your first question is, what in the world is the difference between prokaryotic and eukaryotic cells, and why do you need to know this? Well, you should already know the difference between prokaryotic and eukaryotic from both the last unit and the last lecture. Prokaryotic are single-celled organisms. Eukaryotic are multicellular organisms in the most basic of terms. So, with that in mind, are you prokaryotic or eukaryotic? Well, you're eukaryotic because obviously your body is made of more than one cell. In fact, you have trillions of cells in your body. About 90% of those are not your own cells, they're bacteria that are living with you, but the rest of them are in fact your cells. Now, why do you need to know about this? Why are we taking a whole unit to study cellular biology? Well, two very important reasons. First of all, if you know what your cells are made of, you'll know the vital nutrients they need in order to function, in order to keep you healthy and to keep you alive. And if you know what your cells need, you'll know what nutrients are good for your body. Additionally, cells are what allow you to grow and develop. It is through the process of cellular division, mitosis, that you are able to grow and develop. But in order to really understand mitosis, you need to understand the different parts of the cell. So before we get into cellular division, we're going to talk about what cells are actually made of. And you're going to understand how the cells in your body are different from the cells that are found in the bacteria that are living all over your body. All right, so let's start with prokaryotic cells. They, as I said, are single-celled organisms. They are the earliest and most primitive forms of life on Earth. So we know that 3.6 billion years ago, the first life on Earth was cyanobacteria, and they were prokaryotic cells. They were single-celled organisms. Prokaryotic cells include all bacteria and all archaebacteria. So we learned about the two domains when we talked about classification in the last unit. We talked about bacteria. We talked about archaebacteria being two different domains. Bacteria and archaebacteria are all prokaryotic cells. They're all single-celled organisms that have no nuclei. Pro, that prefix pro, means before. Thus, prokaryotic cells, single-celled organisms, evolved before a nucleus evolved. And they have no membrane-bound organelles inside of them because they are such simple cells. So some prokaryotic cells have a cell wall. Because these organisms consist of only one cell, they need something to protect themselves. So on the outside of the cell membrane is a cell wall, and it's not as rigid as a plant cell, but it provides more rigidity and more protection than just a cell membrane. Okay, that was pretty quick. Well, it's because prokaryotic cells are so simple. Eukaryotic cells are anything but simple. They're pretty complex, and they have a lot of different parts. So fasten your seatbelts. We're going to go on a journey through the eukaryotic cell. All right, so eukaryotic cells are multicelled organisms, meaning they have more than one. They have a true nucleus because that prefix U means true. They have membrane-bound organelles floating around in the cytoplasm. They have a cell membrane in animals and a cell wall in plants. So plants are eukaryotic cells, but they have both a cell wall and a cell membrane. This is a prokaryotic cell. Very basic. We have a membrane on the outside, we have a cytoplasm on the inside, and DNA just kind of floating around in the cytoplasm. That's it. Eukaryotic cells are not only much larger, but they're much more complex. They have to be larger because there's so much stuff. There's this phospholipid bilayer on the outside. We've got a nucleus on the inside. There's some DNA material on the inside. We do have our cytoplasm, right? The three things that all cells have, the membrane, the nucleus, the cytoplasm. Floating in that cytoplasm are all these organelles that are each serving a different function in order to allow the cell to survive long enough to reproduce through the process of mitosis. So your second quick question, compare and contrast pro and eukaryotic cells. So take a moment to pause this video, look through your notes, compare the two, make sure you understand the differences. It's vitally important. Alright, so let's talk about some of these organelles that are floating around in the eukaryotic cell. So that term organelle, as I mentioned before, organelle means tiny organ that's performing an individual function. Just like all the organs in your body each serve a different function, all the organelles in a cell each serve a different function. The endoplasmic reticulum, first organelle we're going to talk about. It's a thin membrane that is not visible under a light microscope. So the microscopes that we have in the classroom will not allow you to be able to see the endoplasmic reticulum. You need an electron scanning microscope to see it. It divides the interior of the cell into different compartments. It's abbreviated as ER, not to be confused with the emergency room. And there are two types of endoplasmic reticulum. There's smooth and there's rough, and they both serve different functions. So here's an image of these thin membranes known as the endoplasmic reticulum, and we have smooth ER and we have rough ER. The rough ER, it's rough because it's studded with ribosomes, another organelle that we're going to learn about. So what does the rough ER do? Well, it manufactures proteins. Remember I said proteins are so vitally important? They're so important that they have an organelle that does nothing but sort them, because there are over 200,000 different types of proteins in our bodies, and they need to go somewhere and they need to do something, and it's the rough ER that gets to manufacture them. They translate RNA. So RNA is what reads DNA. Well, that RNA then has to be translated so it can be made into a protein. And the rough ER is where that translation process starts. So here we have our rough ER, our rough endoplasmic reticulum studded with ribosomes that's translating RNA into proteins. Whoops, sorry. Skip was getting ahead of us. So, what are the building blocks of proteins? You should know this. We've talked about it over and over. Building blocks of proteins are amino acids. It's the DNA that tells the cells which amino acids to bring together in order to create certain proteins. Those proteins eventually create physical structures. So, what you're seeing here in this image, these colors, these are the amino acids, and they are being brought together in order to create a protein. That's a process that you will learn about next year. Alright, so that's the rough ER. Let's talk about the smooth ER. Hey, buddy. Alright, so what does the smooth ER do? It organizes. It lacks ribosomes. This is what it looks like. So the rough ER, it's studded with ribosomes. The ribosomes are actually where the proteins are made, and then they're sent to the rough ER where they're sorted, and then they're sent to the smooth ER where they're further organized. So compare and contrast the smooth and the rough ER. The major difference between the two is that the rough is studded with ribosomes, and the smooth is not. All right, the nucleus. It is considered the information center of the cell. It's spherical in shape, so it's like a ball that's floating in the center of the cell. It is definitely the largest organelle. It is readily visible underneath the light microscopes that we have in the classroom. Centrally located, and it's positioned by filaments. So the reason why the nucleus doesn't move is because it has these filaments that are holding it in place. So here is an actual image of a cell underneath an electron scanning microscope. Again, kind of looks like a fried egg or a poached egg, whichever you prefer. Has the membrane on the outside, cytoplasm on the inside, and then this dot in the center is the easily visible nucleus. Why is the nucleus in the information center of the cell? Very good. It contains the genetic information. It contains the DNA that tells the cell what to do. All right, the nuclear envelope, it's a double layer membrane on the outside of the nucleus. There's that double layer membrane again. That structure of a double layer membrane is really important. It surrounds not only the cell, but it also surrounds the nucleus. And like the membrane that surrounds the cell, the membrane that surrounds the nucleus lets certain materials in and certain materials out. It's pinched together into the nuclear pores that are letting the materials in and out. The outer membrane of the nuclear envelope actually has the endoplasmic reticulum attached to it that are embedded with proteins that serve as molecular channels. So the nuclear envelope very much resembles the cell membrane in that it has proteins embedded in it, and those proteins are allowing certain molecules in and certain molecules out. So it restricts passages of molecules to only proteins and RNA. So the only thing that can get in and out of the cell are proteins and RNA. So the DNA cannot leave the nucleus. So here we have, again, artist's interpretation. This is the cell. This is the nucleus. This is the nuclear envelope that has these embedded proteins, also known as the nuclear pores, that allow proteins and RNAs to travel in and travel out. Okay, ribosomes, definitely the worker bees of the cells. They're very, very busy, those ribosomes, constantly making proteins. Well, not constantly, but a lot, and it's a lot of work. So it's where the RNA is made into proteins. So inside of the nucleus, the RNA reads the instructions on the DNA like it's reading a cookbook, and that RNA travels through the nuclear pores to the rough ER, where it gets sorted, and then it gets sent to the ribosomes, and the ribosomes take the information on the RNA that originally came from the DNA, and it brings together these amino acids and makes proteins. So the proteins are necessary for the function of life. So here it is. This is our nucleus. We have the DNA. It gets synthesized, meaning created, into messenger RNA. That messenger RNA travels out of the nuclear pore to the ribosome, where the ribosome takes the information on the RNA, determines which amino acids need to be brought together in order to create proteins. So why are proteins so necessary? Because everything in your body is made out of proteins. The membrane of the nucleus needs proteins. The membrane of your cells needs proteins. Proteins are what create physical structures in your body. All right, the Golgi complex, delivery. Golgi complex, also known as Golgi bodies. These are individual flattened stacks of membranes that kind of look like stacked pancakes. The Golgi bodies, the Golgi bodies are collectively known as the Golgi complex. They function in molecule collection, packaging, and distribution. So they make sure that molecules get where they need to go. So the Golgi complex is often referred to as the FedEx of the cell. So here we have our nucleus, we have our rough ER with the ribosomes that are busy making proteins, and then we have these flattened stacks that are known as the Golgi complex that are sorting different molecules. All right, lysosomes, often known as the garbage truck of the cell. Isn't that great? Wouldn't you like to be known as